Don't forget to get a wet wipe. Oh, did you? I didn't catch it. Man, I try to be really, really good and catch it every time, but I don't. Okay. And where's my
I don't know why I just like that.
Well, you know, better safe than sorry.
It's fine. We did.
I got my scrubs.
I didn't feel like changing after work today.
Where do you work?
I work at the hospital.
What you?
Phlebotomist.
Oh yeah.
Yeah, I started in June, so it's been some getting used to.
It's been some learning. All right, let's see. I need to see. Well, let me share. And I want to see my chat. Okay, and I'm going to try something new. Because, of course, that's just what I need, right?
I was on my track, but then I confused myself.
Oh, that's all right.
Yeah, I realized that after I did it, I was like, shoot.
Talking about
The lab.
oh yeah, that.
So there wasn't anything we had to turn in, right?
No, you like answered the questions while you were in there.
That's what I thought.
Yeah.
Just wanted to make sure.
Oh, Labster.
Why did I get kicked off the internet?
Oh, Labster. Okay, let's see. Now, where's my flash drive?
Internet problems here. I
Isn't that
don't
what I want?
understand. Like, I couldn't get on my Zoom class on yesterday
Where is
while I was sitting in the library because I couldn't get on the internet.
Okay, so my laptop did the same thing.
Well, I thought I couldn't get
Here we go.
on.
I didn't upload
Okay.
anything.
I think I'm just really unlucky because, like, I had an iPad and the iPad could never connect to the internet ever.
Well, I have unlimited data on my iPad. That's why I tried to do that.
Yeah.
attendance is what I want. Let me try to start attendance. How long does it take Dr. B to set up? Well, everything covers everything else when you start opening things. And I think I try to open too much at once and it doesn't like me. I might, is my audio on? Yes, okay.
It's just my laptop not working.
Good morning. Who's M? All I see is M. Y'all have to put y'all names. It's hard enough I can't see your faces and then I don't even have your name. Good morning, Annie. All right, let's see. This is supposed to be getting big. Here we go. Okay, let me put this down. I'm going to minimize this. And I'm going to start this. Okay, so go ahead and check in. Yes.
Okay, Wednesday when we took the exam,
Yes.
I was here at school, and then I tried to upload the picture of my striper
Uh-huh.
so we can get the Wi-Fi to work. So by the time I finally got home and I tried to do it, it wouldn't let me do a link submission.
Okay.
I left a comment.
Oh yeah, about that. Sorry.
So I want y'all to upload it right there. That's the point of having the submission time in, you know, relatively soon after the exam ends. So if you have to do it, use your phone and take a picture of it right there.
See, I was trying, and like, I can't figure out how to attach the image from my phone in there.
Oh.
I tried to do it on my laptop, and I couldn't get the Wi-Fi to work. So by the time I finally got to Wi-Fi, it was too late.
Okay.
I left the comment on the thing because it wouldn't let me upload it.
It wouldn't let you upload it late. I'll send me an email and I'll extend the due date for you and you can go in and upload it.
Okay.
But send me an email so I remember to do it.
Okay.
And then I'll send you an email that says, hey, the assignment's open, go post it.
Okay. Thank you.
Yeah. So let me answer Austin's. Do we need to screenshot your completion for Labster? No. Dr. Smith knows that you completed it because we have a dashboard in Labster and it tells us what you do and what you don't do and all that kind of stuff. So we're watching. We're watching.
That is the lucky of it. That
stuff. It is. It is. Okay. Oh, there was one other thing I wanted to give y'all.
I keep getting the last number.
I think it's 4.
Okay. Yeah, 1984.
1980, but I didn't know
1984.
I didn't know when.
Okay. I think we're almost ready. We got one more minute. Any other questions before we start class? It was a really long section 1. I don't know what it is about this textbook and section 1. Section 1 is like a marathon for this textbook. It is so heavy. You like Labster? Oh, good. Finally. I mean, like, it took us long enough to get Labster.
It was very easy. Like,
Well,
it was easy to follow.
It should have. That's why we were all excited about it. That's why we worked so hard to get it for y'all. That we thought that it was really good. So I'm glad that it finally. Okay, M. You need to tell me who you are, M. Did you put it in the chat and I missed it? No, M. You need to tell me. Okay, so protein biochemistry. All right, I hate to be the bearer of bad news.
Is this the worst set?
No, it's not the worst. It's not the worst. You need to know your amino acids. There are 20 of them. You need to make flashcards. You need to study your amino acids. I am working on, and I was supposed to be finished with it this morning, and 10,000 other things happened this morning. But by the end of the day, I will send you a link. It's a link to a video that's got some mnemonic devices, and then I put together kind of a Word document that has a lot of different ways to remember them and things like that. So I'm going to send you kind of a study tool. But if you would like to be like the best, nicest person in your class, and you have Quizlet, and you want to make a Quizlet where you have the picture of the amino acid and the name on either side of the card, and then you have like list the hydrophobic amino acids, and you have that on the card, right? If you want to make a flashcard set for amino acids and you send me the link, I will send it to everyone. If you're a kind and generous and nice person and just want to help your friends, I think that's a beautiful thing. So, I don't know. Well, M's got it. So, you need to know the structure, you need to know the name, you need to know which ones are hydrophobic, which ones are hydrophilic, all that kind of stuff. And I'll be honest, I love your textbook, except I don't like the way they do amino acids. And I like to have like one picture and it's got all of them and I can label things. And so I'm going to send y'all today like my version of what I like, and hopefully that'll help you too. Okay, so I'm sorry, it should have been done before class period today, but Dr. B was just not cutting it. Okay, so what I want to try, I know, remember I was having trouble writing on the screen with my PowerPoints and stuff, so I did this a different way. And so I'm going to try this for our clicker questions. So everybody got your clicker ready? You've checked in? If you haven't done attendance, please go and do attendance. We have 41 right now. 42. It's open. It's still open.
Yeah.
Please go and do it.
Yeah.
If you have not done attendance.
I did not know that.
That's why I'm reminding you now. I'm going to give you about 10 seconds and then I'm calling it because we got things to do. And I separated, y'all be so proud of myself, I separated the clicker questions by section, and I have big things in there to help me remember to stop and not get carried away. Okay, so I'm anticipating 48 today. All right, that's good to know. My clicker, Jacob. Try the, do you have the clicker Reef app? I know, you do. Close it down and restart it. My app says reconnecting. Close it and restart it and jump in whenever you can. It's okay. It's not points. It's okay. All right. How do I do this? Here we go. Woohoo! All right. Select a hydrophilic amino acid. Any one hydrophilic amino acid. Oh, I have to start the question. That would be great. That would, yeah, that would help. And I could put the screen on for y'all. That's fine. 6 of 1. F is not an option. Oh, it only goes to E. So disappointed in myself. Well, like it's not like a test where I have to pre-check in the answer. So it could be an option. I'll give y'all 1 minute. It was 48. So I want to see 48 answers. But some people were having clicker problems, so maybe not 48. Okay. Ooh, 49. Woohoo. All right, so when you were looking at this, what you should have said, if we were asking for hydrophilic, is D, well not D, is E or F. So it is F, I know. So these two are hydrophilic. Here, these are hydrophobic.
What's A?
So is isoleucine. Sorry, so is isoleucine. Isoleucine is also hydrophobic. So leucine, isoleucine, valine, they're all structurally super similar, super similar. And so they're all hydrophilic. What makes these hydrophilic? Like what is something that is hydrophilic?
It's hydrophilic, that means it doesn't usually come. It means it readily bonds with water.
Yes, it has a polarity, and so it is able to interact with water. It's able to create those hydrogen bonds, right? And so that's what we want in something that's hydrophilic. So if you look at your textbook, like you could have up, what I would do right now, if I were you, is I would have my textbook open in one panel and be able to kind of look at these amino acids. I can't do them both. I can't manage that many different things. Yeah, or your notes, whatever it is, so that you have them kind of side by side. Okay, all right, Michelle, it's okay. We all have that. All right, so the point is you can attach a hydrogen, right? Like an OH group, something like that is gonna make you hydrophobic, is gonna make you, hydrophilic, and so you're polar. If you're hydrophilic, you're polar. So you want to interact with water, right? Okay, and phenylalanine is an aromatic. Yes, here's the thing with classifying amino acids. Amino acids can be classified in more than one category. They're not just one thing. That's not how biochemistry works. They have multiple functional groups within their side chain, and so they have multiple different functions. And so that's why I like to have, this is like my little, I'm gonna share this with y'all. This is my little cheat sheet, right? It's got all of them on there. And they're grouped in different ways for me. And so I pulled it from an old textbook that I liked. And I'll share that with y'all later today. Okay. All right, so let's go on. All right, so this one is talking about size. So what is the approximate molecular weight of a protein composed of 70 amino acids? You're gonna have to go and remember the molecular weight of one amino acid. Oh, thank you. Numeric. Y'all are keeping me in line. I like it. It's okay. It's okay. It's okay. Just jot it down, see what you get, leave it on your calculator so you can check your answer. That's fine. Okay, 10, well, I guess not 10 seconds. You'll have to calculate. I'll call it a minute 30. But it's not that hard. If you know your conversion, it is a quick calculation.
I mean, if I'm looking at it correctly, well, yeah.
We're going to talk about why this is
So
important.
either this is easy or or Google's lying to me.
You shouldn't use Google. Don't use Google.
Okay.
Don't use Google.
All right. This totally accurate. Totally accurate. Well respected.
All right, what is that? 1
scientist.
minute 30. Put in a response. Tell me what you get. I'm going to call it. All right, so what did we say? All right, so what you should have said was about 7. Now, this might have been a rounding error where you got 7,000 instead of 7,700. What else? I guess it orders them. Golly.
Wow. All right.
So some of them you're off by an order of magnitude, but it depends on how you put it in as either 7,700 Daltons or 7.7 kilodaltons. We typically give proteins in kilodaltons because they're made up of many amino acids, right? So why do we care? What's the point of knowing the approximate molecular weight of a protein?
Because so we know how. So we know how.
Well, remember when we did the cloning, right? We had, I'm trying to write, this was supposed to be better, right? We had our vector and we put in our gene of interest, right? Like we said, okay, maybe this is the insulin gene, right? And we put it into our vector, we expressed it in bacteria. Now, I want to use this insulin in patients, right? How important is it that the entire functional protein gets expressed? Super important. So I should already know the size of insulin. So then I put this plasmid into my bacteria. I tell those bacteria, make my insulin, right? And I purify that protein out of the bacteria. What I'm then going to do is I'm going to run it on a gel. And that gel is going to isolate by size. So it's a size exclusion sort of deal. And you're going to run it, and your protein's going to travel. And depending on size, it's going to travel a certain distance. So something small is going to go very quickly, and something large is going to be towards the top. And you put in, have y'all done these? You've done some. And so you've done it with DNA. So it's a similar thing. You put a standard that you know of certain sizes, right? And you know, okay, this is 7,000 Daltons. This is 8,000 Daltons. So my insulin should end up kind of between the two, right? So what if I got something that was maybe about here? Between 8 and 9. Is that my insulin? No, it's not. It's not. Something's wrong. Something's really wrong. More than likely, you end up with a smaller product because maybe you have extra enzymes in there that are chewing up your protein of interest, or other kind of things. But it's really important to know the size of your protein. Really important in terms of purification and making sure you really have the stuff that you think you have. Any questions on that? Okay. All right. Now we're going to go back to, no, we're still numeric. Okay. So in this one, what you're going to do is you are going to look at, this is a titration curve. So we're adding a base down here, and we're analyzing pH. So we know that there are charges on the ends of all of our amino acids, and some of them have side chains with charges. So I'm asking you, how, knowing the information that's over here, how can you determine the isoelectric focusing point, or PI, of a particular amino acid? And this one is aspartate. I talked about this a lot in the lecture video. I did. Now I want you to apply that. I'll give y'all until about 2 minutes. Uh-huh, I did. I did. But how do you do it? Right? It's true. If you're paying attention, it's easy. That's the answer to everything. If you're paying attention, it's easy. I guess I should turn up the sound in case somebody says something you want to hear. Well, I'm impressed. It's the way to succeed, though. All right, 2 minutes. Click in. Tell me something. The math isn't hard on this one. You just have to know what you're looking for. So y'all click in. All right, I'm calling it in 5, 4, 3, 2, 1. All right, so what you should have gotten was an isoelectric focusing point that's halfway between the two pKa's of the neutral species. How do you know what's neutral? What did y'all pick? Y'all picked 3. We had 3. Oh, we had lots of other things. Okay. So the first thing that you have to do is you have to look at all the different parts of the molecule that can be charged. For aspartate, how many different parts of the molecule can be charged? The N-terminus, the C-terminus, and the R group can be charged, right? So see, we have N-terminus, C-terminus, and our R group are charged. So what we're looking for is the place where the overall, the net charge of the molecule is 0. So if we look here in the highlighted species, we have a plus on our N-terminus and a minus on the C-terminus. So 1 plus and 1 minus end up being 0, right? What's the overall charge for this bottom one? 1 what? Positive or negative? This one's a +1. This one is a with positive and 2 negatives, so -1. And then we have no positive and 2 negatives, -2. So we find the species that's neutral. And then we have to look at the pKa's for those two reactions that get to the neutral species. So pKa1 plus pKa2, and then it's just the average. And so you get 3. Okay. Now, is this scientifically always going to be correct? No, it's not, because you have to look at the environment that the amino acids are in, right? They're never just in water in solution all by themselves. They're really not. But that's what this number is. This number tells us just basically by itself in water, this is what the isoelectric focusing point would be. But it doesn't happen.
How do you know PK1 and PK2?
You have to do a titration curve to figure out what the pKa's are.
No.
They were given, the pKa's were given. See, pKa2, it's 3.9, pKa1 was 2.1, and so those are the numbers we put in here.
No, I know that one. How do you know PK1 and PK2 are neutral?
Because they're the two pKa's that flank the neutral species. So these two pKa's control the kinetics of the reaction that form the neutral species.
Okay.
Okay. All right. Okay, so you're really not going to have to H. Like, I didn't even think about this. So you can put a short answer. It's short answer. It's not, Hannah, no, it is not always pKa1 and pKa2. You have to look for the neutral species. So you would have to be given this and look for the neutral species. Okay. All right, so I put this one as short answer. So you can give me more than just A through E. All right, so the side chains of some amino acids are charged at physiological pH. Select an amino acid that possesses a charge on their side chain at physiological pH. Because we're always trying to think about what's happening in the body at physiological pH. And what is physiological pH? But no one's going to answer. Anybody? Physiological pH? Yeah, it's around 7. It's around 7. It depends on what system you're in, but around 7. Yeah, 7.4, 7.2. Just depends on where you're at. All right, come on, y'all. We need those flashcards. Y'all need those flashcards. But y'all have studied a little bit of amino acid.
I'm taking cell biology right now, and both of these classes are under the exact same thing. The only difference is cell bio groups a little bit.
Yep, yep.
Like we have to know the negative charge and the positive charge, and that's 2 groups in cell bio.
Well, you do, but you do have to know it for this, so
Yeah,
that works.
But I'm learning it twice, so it's fun.
That's good. All right, I'm calling it a minute 45. Take a guess, y'all. Take a guess. We've never had to know the structure. That's true. Okay, you've never had to know the structure, but you're familiar with the terms. Yeah, you're gonna need it for the MCAT. You're gonna seriously need it for the MCAT. And you're gonna need it like the next like 5 chapters are protein chemistry. So if you don't know your amino acids, it is gonna bite you. It is really gonna bite you. Okay, I said I was gonna call it. I'm calling it. Okay, so you could have had multiple answers. There were multiple correct answers here. So we're looking for charge on their side chain at physiological pH. Let's see, oh my gosh, I didn't think about correcting these. Oh, and y'all put some put multiple. Okay, wait, K, and then it was B. I was like, is that A or B? B's right. So B, F, and K, yes. B, F, and K, F and B, B and F, F, not E. Okay, I think I got all the right ones. I think. All right. And proline. Okay, so if you have a charge on your side chain, right? That's in the category of our charged, but you have to understand, is it going to be positively charged? Is it going to be negatively charged? And how do you determine that? What are you looking at? Anybody? It's kind of in the question. Like, is aspartate always going to be positively charged? Is it always going to be negatively charged? Why do we classify some as positive and some as negative? Yeah. What is it based on? At the physiological pH. So depending on what the pH is, each of the side chains has a pKa value, like what we just did, right? So what happened when we titrated in sodium hydroxide? What happened to that charged amino acid side chain? It was either protonated or deprotonated, right? Okay, so in order to tell what you're going to be, if you're going to be positive or negative, you have to be given a pH. If you are not given a pH, then you have to assume that you are at physiological pH, which is 7. So that's why when you look at some of these, right, like here, I'll show you, it'll tell you which ones are acidic, which ones are basic. Well, how can you determine that? You have to assume that this table was given to you based on physiological pH, okay? So you have to understand why we're calling them positive, why we're calling them negative. It's because of the protonation or deprotonation of the side chain. Good. Okay. Now we're going to apply that. Okay, but first we're going to do this. All right, same thing. Side chains of some amino acids are aromatic. We only have a few aromatics. See if you can select the aromatics. And brownie points to anybody who can put in the chat the definition of an aromatic. In words, not in pictures. And when coronavirus is lifted, I'll make brownies. I can't do it now, it's against the rules. It's a alternating double bonds on a ring structure. That is correct. Why do we call it an aromatic?
Because it's giant. Oh, because it's giant involved. Because it's generally involved with nice smelling.
It smells. That's why we call it an aromatic. And it just so happens that it is a double bond on a ring. It's alternating double bonds. So double bond, single bond, double bond, single bond. That gives you that characteristic. So Jaslyn, you get the brownie points. So I guess she gets first pick on brownies. That should be it. All right. So let's see what we got. So there was only one correct answer choice, and it should have been H. And most people got that. Oh, wait, somebody typed in tyrosine. Yeah. Okay, so those should be my correct answers, right? So you need to go in. This is one of those ones that you need to go in and make yourself the flashcards and start studying them. I mean, that's just sad to say, but true. Okay, so now we can do multiple choice. So which form of cysteine is oxidized? Your answer was H. What was H? Did I mark H? Ooh, ooh. Oh, I can't go back. Can I go back? H. Is there another H? Oh, it was H period. That would be it. Okay. Can't, oh, I'm sorry. Can't sum with resonance of lone pairs. You do have resonance of the alternating double bonds. So the double bonds, it's not really a double bond in a single bond. It's really like a 1.5 bond because of the resonance in all those single and double bonds. Courtney, is that what you were asking? Okay. All right. Come on, y'all. A or B? Which one's oxidized? What do y'all use over here? What's your oil rig? Okay, that makes sense because we're Louisiana, right? Oil rig. What does oil rig stand for?
My professor's always told me Leo the line goes GER.
Yeah, that's the other one. Leo the line goes GER. Either one.
They also taught me they also taught me does tag give these cookies out for good.
Okay. So if you look here, right? If we look, I even highlighted the residues that were important for you, right? How many bonds and how many electrons are here versus over here, right? We have fewer electrons in one sulfur-sulfur single bond than in one bond here and one bond here between the sulfur and the hydrogen. Yes, you are losing electrons. Yes, so that is oxidized. So this is really cool. Cysteine is really, really cool in the formation of really large proteins, right? Because we know in eukaryotic cells, a lot of times you make subunits, right? And then the subunits have to be linked together. Well, how are you going to link them together once they're already formed? This is a really great way to kind of lock those components in together by our disulfide bond or disulfide bridge. Sometimes you'll hear it that way. Okay, good. All right, this one is going to be short answer. Which amino acid is the smallest and least chemically active? Smallest and least chemically active. It is the only one that is non-chiral. Well, yeah, the least, smallest and least chemically reactive. But smallest should give it away too. Same thing, smallest and only one that's achiral. Giving y'all till 1 minute and then I'm cutting it off. Five seconds. Okay. So what did we say? Oh, I have them out of order. No. Oh man. Glycine is correct. Glycine is correct. So when we talk about the structure of glycine, right? What's the general structure of an amino acid? Right? We have an R group bonded to our carbon, and what's our carbon? What do we call that carbon? We call that the alpha carbon, right? And then we have it bound to another hydrogen. So sometimes it's just written CH that way, right? So it just depends on what book you're looking at. And then what do we have coming off of each side? An amino group, so this is the N-terminus for positive CH3, right, if we're by itself. And then we have a carbon, and that carbon is double bonded to an oxygen, and then another oxygen, right? So if we look at glycine, what is the R group on glycine? It's just a hydrogen. So this is the only one that's achiral. All right, and that makes me really, really sad that I put something out of order. Like I don't want to show it. It makes me so sad. Oh, no, I didn't. Okay, I didn't. But then I just did show it. I just didn't put the answer for that one. Okay. All right. So this is super important for your lab this week, right? This is what you're doing in lab this week. So what I have here for you is a graph of wavelength versus absorbance. So what is similar between phenylalanine, tryptophan, and tyrosine? They're all what? They're all aromatics. So aromatics can absorb at around 280, right? Phenylalanine is a little bit lower on the wavelength scale, but you can look at 280. So when we look at 280, what two amino acids are we actually looking at? Tryptophan and tyrosine. So you can give, if you know the sequence of your peptide, and you know this information, you can calculate what your absorbance should be and then use that to predict concentration. So I want you to calculate the absorbance of our peptide. So this is the molar absorbance. So I give you a peptide, you calculate what the absorbance value should be. All right, I'm calling it at 1:15. No, I'm sorry, 2:15. 2:15. When I say that, like the number always jumps of people who have submitted. Okay, so what are we looking at when we try to answer this question? Before I look at my answers, how am I going to calculate it? I need to look at 280 and I know I see tyrosine, and I know I see tryptophan. So I'm going to look in my peptide. I'm going to look, okay, if I have tryptophan here, it should have a molar absorbance of what?
5,000.
About 5,000. So how many tryptophans do I have? 1, 2.
So 10,000.
Okay, then tyrosine. Tyrosine's about
1,000.
1,000. How many do I have?
1.
1. That should be my calculation to find my molar absorbance. So I think we just lost a 0 on that one. Yeah, it happens. It happens. Yeah. Yeah. So if you know your structure, of your peptide, you can use that to make some predictions about your molar absorbance. And sometimes you do and sometimes you don't. So you can't always use that method. Okay. Oh, this is, yeah. This is going to be a short answer. All right. I want you to tell me the amino acid sequence of our pictured tripeptide from N-terminus to C-terminus using 3-letter abbreviations. So you're going to do like A, B, C, dash, E, F, G, and it's really lowercase. It's really not. Those aren't uppercase. B, C, E, F, G, H, J, like that, in that format. And did I make it go? I didn't make it go. Have to make it go. I was really nice to you for this one. I even highlighted the side chains. Your rings, if you learn the ones with rings first, that's going to help you. You'll be able to quickly figure those out. But just because it's a ring, does it make it an aromatic?
No, it's not automatically a aromatic.
Okay.
A ring is just one of the, it's just, it being a ring is just one of multiple like guidelines that has to fulfill or
To be an aromatic,
or
that is
what
correct.
word I'm looking for, like not guidelines, but
Requirements.
yes, requirements.
Mhm.
Thank you.
All right, y'all. Come on. 2:45, I'm calling it. Unless people have left the Zoom, which wouldn't be nice. Be nice if I could make y'all look. Oh, Annie, we have Annie's dog.
And she did ask if she wanted her dog to attend.
She's there though.
She's there.
Yeah, she did ask if her dog could attend.
I don't know how I go through and see.
Good luck.
Yeah, more
Good old
people.
Labrador.
See the arrow?
Oh yeah, there it is.
Good old Labrador.
Nobody wants to put their picture up. I don't blame y'all.
mean, I mean,
Okay.
I don't want identities. I don't want
3:15. All right, what do we got? So what should you have gotten for your amino acids? You should have gotten, oh.
I mean, he's not wrong.
Oh my gosh. Okay, let's see. Do we have any correct answers? Y'all, it's like open book though. This is open book.
I mean.
Well, we
if you have
started his.
I didn't study yet.
Yeah.
I don't know what's going on.
Say that again?
I didn't study yet, so I don't know what's really going on right now. I'm sorry.
Oh, y'all know y'all supposed to watch the lecture and read before you come to class, right? Like that.
I was just mad from the exam. I'm sorry.
Oh, you were mad from the exam? It wasn't that bad. All right, so what was really cool about this one is that I gave it to you in the right order. So I gave you the N terminus to C terminus, right? So we have asparagine here, histidine, and then arginine, right?
I got histidine at least.
So you need to be able to identify in a chain like this. You also need to be able to identify where is my peptide bonds, all those kinds of things you need to be able to find, right? You don't have to memorize them all for today. No, you don't, but you can have a textbook next to you. Your notes open next to you. It's in class. I'm not quizzing you. It's hard to find time. I know it's hard to find time. I know. I get you. I totally get you. Okay, but it's open book right now. Use your book. Use your resources.
Still a pretty big book.
It is a pretty big book. Oh, Ethan's working on the Quizlet. Help is on the way. All right, so we know that when we talk about amino acids, right, it's all of really the different side chains that give our proteins their properties. It's not really about the backbone. It's really about the side chains, right? And so when we do this, now every textbook's going to divide them differently, but your textbook, what 4 categories does your textbook separate them into? You're going to be blessed, Ethan. Yes, Cameron. Yes, Cameron, go. Yes. Yes. Not all heroes wear capes. But yeah, this class is a fast-paced class. It really is.
I've noticed.
It is.
No, exactly. Yeah, I know you feel like.
You just
Rather
need
say
the
so,
amino,
like
I would say
trans-
amino acid structure and name, and then classification, and like any of the special characteristics, like which ones contain sulfur, which one's achiral, you know, things like that. So everything, yeah. Which ones can be phosphorylated? That's another good one, right? If we're talking about groupings, but groupings that are not in your textbook. Yeah, fun. Yay. Okay. All right. So we should be about, I don't know. I don't know. I don't think M's ever told us.
When in doubt, C. Always pick C.
When in doubt, pick C. Not always. But no, when in doubt, always pick the same one. That's the mathematical right way to do
I
that.
know exactly.
If you're taking a standardized test, it's okay, Julie. If you're taking a standardized test, I'm not saying that C is the most likely answer. What I'm saying is pick one answer choice, and for the whole test, use that one answer choice. If you have no idea, if you can eliminate things and you can eliminate that answer choice, eliminate it. But that's the mathematical, statistical way. Okay, so you should have said charged, aromatic, and then they call it hydrophobic and hydrophilic, but you could have said in terms of hydrophilic, polar, uncharged, or hydrophobic, allopathic, right? So there are different ways that you could have said that, but those are your basic classifications. All right. Okay. So, of all the different bond types found in proteins, right? So now we're talking about a peptide, which has a partial double bond character.
Partial bond. Does that mean like?
Partial double bond.
Does it mean like the second one just like gave up halfway and grew inside?
No, we talked about this earlier today in this class period. We talked about it. We talked about the property that leads to a partial double bond, not to the location of it, but we talked about the property. I think this one. I bet you could get it if you look at a structure.
I think to find the structure then.
I always like to print out my notes before I came to class. Always worked out pretty well for me. All right, I'm calling it. So 5 seconds. Like hoping other people will click in. Okay, so what did you say? You should have said C. You should have said the peptide bond, right? So if you look, here's a standard peptide, right? This is our alpha carbon. This is our carboxylic carbon right here, right? And then it goes into the nitrogen of the next amino acid. So if you look here between your carbon and your oxygen, what do you have? A double bond. Well, that double bond can actually move and go between the carbon and the nitrogen. So when you actually measure the distance of the bond lengths of the actual proteins, what you're going to see is resonance. So you have not really the length of a double bond and not really the length of a single bond here. You have something in between.
Wait, shouldn't the bond on that? Oh wait, never mind. It's the end of the game.
It's the nitrogen.
Okay, the nitrogen.
It's the nitrogen. Yeah. All right. Now, because of this causes a particular characteristic of proteins. So the next question is, what is the consequence of this partial double bond? Right? We know we have a partial double bond, but what's the consequence? So I'm giving you this little reminder because hopefully you've at least watched, right? But you have angles on either side of the peptide bond, right? And the angles are called the phi and psi bonds. So I want to show you this. Here's our peptide bond. Here's our psi bond, and here's our pi bond, phi bond. Say it right. So when you're looking at the answer choices, what we're asked about is comparing the peptide bond and these psi and phi bonds, okay? So when you look at your answer choices, I want you to be thinking about that. Now that you kind of have it on your screens, does everybody have it on their screen? I want to switch over so that you can look at the picture. Is that okay? Because you have the answer choices now. Okay, I'll give y'all 10 seconds. Okay, so what do we say, right? So let's go back to our answer choices. Whoop. So what do we say? Oh, good. Very nice. Very nice. Okay, So, right, what is this telling us? It's telling us that if we have a double bond here, can you rotate when you have a double bond? Nope, you can't rotate. But what if you have a single bond between that alpha carbon and the next carbon? Can you rotate? And what about between the nitrogen and the alpha carbon? Yep. So when you think about protein folding and protein interactions, yes, the peptide bond is pretty much locked, but the side chains can move around because you have rotational freedom around the alpha carbon. So a lot of times what we'll do is, right, if we know that somebody has some sort of, did I mark it? Yeah, okay. Say they have some sort of mutation, right? And we change from one amino acid to another amino acid, right? We usually have a crystal structure of the normal wild type protein, right? So what we can then do is model in, okay, here was our normal residue. Now we put in our now mutated residue in that place, and then you have to kind of move it around and see where it could fit. Oh, if it's over here, it's interacting with something it shouldn't be. So it gives us a way to think about how mutations actually lead to physical diseases. If we understand how those side chains can move about the molecule while keeping that peptide bond locked. So we can mathematically apply that. Anybody done any of that yet? Some people working in labs? High mole, anybody? I guess that's a no. Okay. If you do research, if you do undergraduate research, you might actually do that. Okay, so it should have been B. We said that. Okay. So and you do have to E. Okay. So we're good on that. All right. So for the following charged amino acids at pH 7, tell me whether the net charge on the amino acid is positive, negative, or neutral. Oh, I forgot. This one you can't really answer on clicker. You can't really answer on clicker because I just got carried away. But look at them and guess. Look at them and guess. I'm going to give y'all, I don't know, 2 minutes to kind of look at them. I'm not asking for the charge on the side chain. I'm looking for the net charge on the amino acid.
So how would you write this down in like nature?
I would write like A, positive or negative, B, positive or negative,
Okay.
C, positive or negative,
Okay.
or neutral.
Okay.
So if you wanted to jot it down, A, this, B, that, so on and so on.
Okay.
Hint, hint, you might need to take into effect the pKa's, right? Zach's trying to help everybody out. Anybody want to take a guess on our negative ones? Put them in the chat. See, Zach's just got everybody. All right, so what about E? Well, I'll argue with neutral. I'll argue with neutral. Okay, so when we look at this, right, our amino group, we said, has a pKa of 9.7. So at pH 7, what's going to happen to that amino group? At pH 7, our pKa is above the pH, so it is going to be protonated, right? Carboxylate group, pKa is 2.3. So we are below the pH, so it is going to be deprotonated. Now you have to look at the side chain pKa's. So if we look at the side chain pKa's, oh, I did that, okay. Right? If we looked at A and B, both aspartate and glutamate, right? These have pKa's of 4.0. So what's going to happen to those is that they're going to lose their hydrogens. And so now we're going to call it aspartic acid and glutamic acid, right? So if they have those hydrogens, that's why we call it the acid, because in the base form, they've deprotonated. So then if we look at our next two, right, lysine and arginine, their pKa's are above the pH. So now their side chains are protonated. That's why when you look at this book in the textbook, the one that is circled is the one that is going to be present at a neutral pH or at physiological pH. Now, the last one we need to talk about is histidine. So if we look at histidine, Ethan, Kayla, yep, y'all are right. If we went by exactly 7, we would say it is positive. But here's the thing about amino acids. Even if I put them in an aqueous solution like the cell, and I said, okay, the cytoplasm of the cell is at pH 7, it doesn't necessarily mean that the environment around histidine is pH 7. The local environment around histidine makes a really big impact. And so histidine is really cool because it can function both as a hydrogen donor and as an acceptor. So technically, if you follow the rules, this would be positive. But be careful with that. This is one of the ones that I would like circle and I would put on the flashcard, right? That its pKa is really, really close to that of a neutral pH. And so it can kind of function either way. So its environment determines its charge. So if I ask that on the test, I wouldn't ask it that way. I would ask it more like, which amino acid can act as both a hydrogen donor and an acceptor at normal physiological pH, right? Something like that. Okay. All right, now, due to its partial double bond character, the peptide bond restricts rotation. We said that. And so it constrains all of our atoms in that backbone to lie within the same peptide plane. So rotation in the peptide backbone is around the 2 bonds that flank the adjacent what atoms between which the peptide bond is formed. So between what 2 atoms can we rotate? Class ends at 10:40. Oh, thanks, Cameron. Class is over. I'll give you this answer. Can you relate the relationship between pH? You can, Jacob, but it's just easier to look at the average of the pKa's that control the neutral species. Like, don't make more work for yourself than you have to. Okay, so I'll give this one, this is your alpha carbon. This is your alpha carbon. Oh, y'all are changing. Haha. Okay, so y'all are good. I'm sorry, I was not even watching the time. Y'all have any questions? Stick around. Annie, please bring your dog again. Y'all have an awesome weekend. Go watch the videos, go read your book.
Dr. B,
Yes.
do you know around when test grades will be posted?
As soon as I finish grading them. I'm working on it. I am working on it.
Okay.
I am working on it, I promise. But it's not all multiple choice.
We appreciate all of your hard work.
Oh, thank you. What time is Labster due today? Midnight. I don't think there's even a way for us to today. So as long as you submit it today, we're good. We're not doing it this weekend class. We are not doing it this weekend class. It's just regular.
Wait, have we always been having?
No. We have 2 class periods to make up. We're not making it up this weekend. We'll make it up like the following and the one after that.
Like, is it like the week after?
All it is just an extra lecture. But I'm not giving y'all an extra lecture the day, like the weekend after your exam. I feel like that's not a good thing.
Like hosting a lecture and is still doing something on Saturday class. Yeah.
I'm sorry.
Yeah.
You guys have a quota and everything. Let's seriously, this is ridiculous.
Well,
I'm not happy whenever he said that. I was like, you made us watch a 3-hour lecture at home, and you're still gonna do a Saturday class? Yeah.
Like I
oh,
said,
that's terrible. Okay. Anybody got any questions on the chat? Anybody still
I have
on?
a question, Dr. B.
Yes.
Can you go
Tristan.
back to the histidine thing?
Can I go back to it? Yeah. Ooh, here we
Okay.
go.
I'm confused because I thought if you increase the pH, it's closer to a basic environment, which will deprotonate histidine and make it negative.
If you are,
Have a good day.
you
Like
choose.
if the pKa is 6 at this point, and then you increase the pH of the environment to 7, then won't it deprotonate to
You're
make it
above, you're positive, so you should be negative. You're right. This should be negative.
okay?
I can't change that. This should be negative. You're
Okay.
right. This should be negative, but can be either positive or negative. Yes, the final answer should be negative. I'm sorry. My
Yeah.
bad.
Okay.
Yeah.
Histidine can fluctuate, but like it'll kind of tend towards the negative side.
Yes.
Okay.
If it is in a pure, if the immediate environment around histidine is pH 7.
So a difference of 1, I guess, unit from pKa to pH 7 is not that significant. So like how big of a difference would it be before it like stops fluctuating and like takes a more permanent formation?
It's not going to be permanent.
Well, yeah, but
Wait, ask me that again?
like how high would you have to raise the pH for histidine before it pretty much stays in a positive formation more so?
I don't have an exact answer for that.
Mm-hmm.
Because your local environment fluctuates so much that you typically don't stay in that high or that low of a pH. So histidine is a good amino acid that will allow differences based on acidic or basic environments. So like if you have something that is pH dependent, if you have a reaction that's pH dependent, you can control it through histidine residues. Does that make sense? So that you can go back and forth. As the pH changes, you'll get an effect because you're either protonated or deprotonated.
Okay. Thank you very much. I just wanted to ask that question.
No, that's fine. That's fine. Absolutely. Anybody else?
Okay, how you have it written like right above the pKa of histidine, where it says the pH is below pKa, then the side chain is protonated. Is that kind of like a rule, I guess you could say?
Yeah, yeah. If the pH is below the pKa, the side chain is protonated. Yeah.
So like if it's the opposite, then it's...
So if it's the opposite, so if the pH is below the pKa, if the pH is below the pKa, it's protonated. Is it protonated? The pH is 7 and the pKa is 10.5.
Yes.
Protonated.
Okay, I just wanted to see if that was kind of like a rule, because I was kind of still confused by that.
Yeah. Okay, anybody else? Annie, what's your dog's name?
This is Drake.
Drake. Oh, look, he turned and looked. He's very handsome.
He appreciates that. Oh, he's yawning. He's not interested in class anymore.
Yeah, you know, it was a long class. Well, y'all have a great day.
All right. Yep. Y'all have a great weekend.
All right.
Wait, say bye-bye. No? Okay.